Several million mild traumatic events occur in the United States annually, but they often go unreported due to their lower severity. Most people hear about traumatic brain injuries, but general mild trauma does not get as much attention as it should, especially considering it occurs more frequently in regular everyday life, such as falling down the stairs or being hit in a car crash. There are still a lot of issues that can result from someone who only received mild trauma, but it is not yet clearly understood. When an injury happens, programmed cell death occurs, and this is critical for both health and development. In apoptosis, a process of programmed cell death, it either occurs during development to eliminate unwanted cells, or it is used to rid the body of cells that are damaged beyond repair. A cell surface receptor, called Draper, is required for the recognition and engulfment of apoptotic cells. However, in our lab we have Drosophila, which are fruit flies that are missing Draper in their bodies. These are known as mutant drapers, and they can lead to a lot of inflammation and tissue damage. To study cell death and clearance in mild traumatic injury, the ovary and gut of the Drosophila were observed. Since Draper mutant Drosophila don't have the ability to remove their dead cells through that certain receptor, they're left with a lot of damaged cells. This can cause complications to the body, and in a previous experiment done by Professor McCall, it was seen that these Draper mutant flies had a lot of neurodegeneration. In our study, we're focusing on female response due to the lack of information on it. Females are capable of reproducing and are extremely important to keep healthy in order to keep the circle of life going. Therefore, we studied the egg-laying process and dissected the ovaries of female Drosophila after trauma. In their ovaries, we knew that if they held on to stage 14s, which are fully fledged egg chambers, that they were not as healthy because typically these stage 14s should be laid and not withheld. In the regular white 11-18 flies, which served as the main control, the flies had a predictive relationship where the traumatized flies laid less eggs and held on to more stage 14s, which turned out to be true. However, in the Draper mutant group, the traumatized group actually laid more eggs and held on to less stage 14s than the control. This was a shocking discovery as it has not been seen before in prior experiments. This discovery is crucial to keep researching as it suggests that even with the absence of a cell surface receptor for damaged cells, the body was able to recover to a more biologically healthy state. This can help in the future for people with a lot of conditions such as rheumatoid arthritis, cardiovascular disease, Alzheimer's, and many more in which dead cells are accumulated. Now we can better understand the effect on female physiology after trauma, as well as the unknown protective mechanisms the body performs when missing critical components for getting rid of cell death.